Now that we know what a clinical trial is, let's ask why. Why do we need this rigorous testing process? It might seem obvious that we should test medicines before giving them to people, but the framework we have today didn't always exist. It was built, painfully, in response to catastrophe. The most sobering example is thalidomide. In the late 1950s and early 1960s, thalidomide was widely prescribed across Europe and in many other parts of the world to help pregnant women manage morning sickness. It was considered safe. It was sold over the counter in many countries. What happened next was catastrophic. Thalidomide caused severe birth defects, a condition called phocomelia, in thousands of children born to mothers who had taken the drug. Children were born with malformed or missing limbs. Estimates suggest more than 10,000 affected children were born worldwide. The tragedy exposed a fundamental gap. The drug had never been properly evaluated for teratogenicity, its ability to cause harm to a developing fetus. There was no rigorous regulatory framework to catch this before the drug reached the market. In the United States, a scientist at the FDA named Frances Kelsey refused to approve thalidomide because she was not satisfied with the evidence on its safety. That decision, widely regarded as one of the most consequential in FDA history, spared the U.S. the worst of this global disaster. The thalidomide tragedy led directly to major legislative reform. The Kefauver-Harris Amendment of 1962 required pharmaceutical companies to prove not just safety, but efficacy, that a drug actually worked before it could be approved. It also established the requirement for informed consent before any patient participated in research. A second landmark and deeply troubling case is the Tuskegee syphilis study. This was a study run by the U.S. Public Health Service from 1932 to 1972 for 4 decades in Macon County, Alabama. 399 Black men with syphilis were enrolled. They were told they were receiving treatment for bad blood. In reality, they received placebos. And when penicillin became the established cure for syphilis in the 1940s, it was deliberately withheld from these men so that researchers could continue observing the natural progression of the untreated disease. The study was exposed in 1972 when a whistleblower, Peter Buxton, leaked the details to the press. By that point, 28 men had died directly from syphilis, 100 more had died from related complications, 40 wives had been infected, and 19 children had been born with congenital syphilis. Tuskegee fundamentally changed the ethics of human subjects research. It led directly to the Belmont Report in 1979, the foundational document of research ethics, which established 3 core principles: respect for persons, beneficence, and justice. It also led to the creation of institutional review boards, the ethics committees that must approve every clinical trial before it begins. These tragedies, and others, are the reason that clinical trials today operate under a rigorous ethical and regulatory framework. Today, every clinical trial must have a protocol reviewed and approved by an independent ethics committee. Every participant must give written, informed consent. They must be told what they're agreeing to, in language they can understand, and they must agree freely. Regulators must authorize the trial before it begins. Safety data is monitored throughout, and every serious adverse event must be reported promptly. The framework isn't bureaucracy for its own sake. It is a direct response to what happens when human subjects research is conducted without adequate safeguards. Let's summarize Lesson 1.2. Clinical trials are necessary because human physiology is complex, because medicines can cause serious harm, and because history has shown us, repeatedly, what happens without oversight. The regulatory framework we work within today was built on the lessons of real tragedies. Understanding that history makes you a better, More conscientious clinical research professional.